I'm going to talk very pragmatically, yeah, about diagnostic and sort of how I've experienced working in the diagnostic field in research and diagnostic, but also in non-academic settings to work on diagnostic and really work towards global health impact. And let's see what I've learned in that context. So I've tried to set this up a little bit across three spheres. Really, you see, like from quite early on in my career, maybe, well, not that early, but yeah, working more really lab-based in on multiple studies, but all sort of around this question, like what is actually making people sick? Because if we want to think about diagnostic, we really have to, target the diagnostics to the population, to the problem, and really understand what is making people sick. So what do we really need in terms of diagnostic? And then gradually, from being sort of really deep in the lab, I kind of moved to a more non-lab based position. I worked with international organization FIND in Geneva, and kind of sort of that broadened my horizon a lot to what other kinds of evidence is really necessary to move diagnostic forward and to think about impact in the context of diagnostic. And then sort of the last, maybe the last place, which is kind of connecting all of this learnings and all of the takeaways and seeing how do I connect that now by really thinking about infrastructure, technology as part of a system and not as the core solution, maybe. So let's see. And I hope I can take you on a journey. I have a little disclaimer. So, I picked studies right now for this talk that are a bit illustrative. These are not maybe the most exciting, and maybe these are not the ones others would have picked, but I think they illustrate nice how this evolvement from really thinking about very deeply in the lab, what is making people sick, and how do we need to improve the testing to kind of the sort of bigger picture thinking. So I kind of picked a couple of things. I put at the end my you can have a look at other publications. And by all means, if anything interests you in more detail, reach out to me. This is a very early picture of me in Southeast Asia and Laos when I worked for Charité. This is sort of baby Sabine writing into a very big laptop, and kind of shows how old this picture is, because that laptop was like very, very big and thick. And working on malaria diagnostic here. Yeah, so let's start and stay in Laos. And I kind of said sort of the theme of the first bit is about like what is really making people sick. So I worked with the University of Oxford in their research operation in Southeast Asia. The logos that are on these slides are really just looking at like where was I at the time. It's not necessarily all the set of collaborators. I want to be very transparent. It's only where I was at that time, just to give you a bit of an idea. So yeah, the first question that really interested me was what is making people sick? You saw the previous slide, and there it was all about malaria. That was also in the same country, but at the time when I came back to Laos, because of a lot of efforts, malaria wasn't that much of a problem anymore. So the question really was, okay, these people still are sick. What is making them sick? And this is what we really spend a lot of work and a lot of time collaborating, within. I was the lab person, if you want, as part of a multidisciplinary team with lots of clinicians and clinical trial experts. Here, this was a study looking at what is causing central nervous system infections, what bacterial causes are contributing to that. And the major finding here, which was very unique and at the time, was that it wasn't what we thought it would be, but it was doxycycline responding pathogens, or a lot of rickettsial pathogens, sort of a group of pathogens that is widely under-recognized, which is what I learned then and what sort of was contributing a lot in Lao then to changing of treatment plans, not necessarily for central nervous system infection, but for other infection. So this doxycycline wouldn't be the first line treatment otherwise. And this was kind of a really important finding that 35% of the infections were caused by that. So that kind of really um, yeah, it was to me really interesting, and it was interesting to everybody because we used really high, we used molecular diagnostic. We had retrospective samples. We used like a combination of different types of tests, something that isn't possible in routine. And something that's also not possible for surveillance. So a lot of my time at the time was also spent like tweaking and playing around and thinking about unique and maybe alternative ways of improving our understanding of this group of pathogens that you cannot normally grow in a blood culture. So one of these pathogens, these rickettsial pathogens, is called Orientia tsutsugamushi. Quite a fun thing to say, but not a fun thing to have. So as you might have seen, that was also one of the major contributors to these central nervous system infections. So it was important for us to understand better how big is the distribution. So one thing really development of new tools, like thinking out of the box is like, this is an illustration. Was an illustration at the time of how can you think maybe creatively of improving our knowledge of what is out there. This was a methodology we played with and published in the end to use leftover specimens from hemoculture, from blood cultures. They could be used, and we could show that there was growth of these pathogens that wouldn't normally grow. And that was kind of cool. And I know, and I always wanted to explore if that was really a way of seeing a little bit more into the epidemiology of that pathogen and that group of pathogens. And some colleagues are currently, I think, exploring that in Southeast in Chile in South America. So who knows? Maybe it works, maybe it doesn't. But in the lab, it did work. But maybe how much does that work in reality is maybe where we can talk later, because we didn't test that at the time. That was really thinking just about novel ways and coming up with something that works in the lab. One thing that we also, as part of that work, really explored was, well, are these pathogens? Traditionally, they're sort of thought to be only in Southeast Asia, but I was still in touch and I was working with my old colleague, Thomas Meitzel, who was my colleague at Charité when I worked in Berlin. And he worked in Chile now, and he was looking at rickettsial pathogens and febrile illnesses in Chile. And we started to talk and we started to collaborate. And we, in our lab in Lao, were able to confirm that they actually had Orientia tsutsugamushi-like strains in South America. That was like worth a New England Journal of Medicine report because that was completely not described previously. So again, this question, well, if we don't know what's making people sick, if you don't know the causes, it's very hard to really diagnose them appropriately and then treat them appropriately. So this was kind of maybe a nice story at the end, like when I left Lao to round this up after many years. But one thing that stuck with me was like we had this beautiful Oxford funded, well, Wellcome Trust funded laboratory. We had a lot of capacity. We could do sequencing. We could do a lot. That was not the case in many other places. And so these pathogens, but also other things like that really need diagnosis, cannot really easily be diagnosed if you don't have that capacity. So that really inspired me at the time to move on to a role that wasn't academic anymore, but was at FIND and the Foundation for Innovative New Diagnostic in Geneva. And that's when it became a lot more strategic, a lot more maybe outside of the lab, and a lot more thinking about, okay, how can we target and how can we develop diagnostic tools that actually fit the needs of the majority of the people that need to use it, that don't have the fancy cool labs, and that can't send reference sample maybe, but really need to test where they are. So what is it that we need? And in this case, the example here is, there would be others, is about differentiating bacteria from non-bacterial diseases to target antibiotic prescribing. But really, what was new to me was this sort of strategic thinking around like target, what does the profile of the test need to be to really be useful to the population that needs to use it? Where do they need to use it? Who's using the test? How long can it take until you get the results back? And all of that is called target product profiles. The WHO, World Health Organization, uses that a lot, but also funding agencies and other international organizations, but also companies use that to inform what do they actually need to develop. Because ideally, we're only developing things that are actually are used and that are impactful in the places where we think they're needed. So the methodologies here were quite different from what I knew from the lab. It was really about gathering evidence from experts, getting consensus around a topic and what is it we need, and then sort of use that to inform the developers, the funders, the clinical trialists that this is where we should focus on. This is the area we need to work on. These are the target characteristics that the test needs to have to really be impactful. And then as a consequence, this is also what we should be looking at when we trial or evaluate tests. Like, what is it that really is important? Like, who's the population that it needs to be tested in? Because that's defined, we defined in the target product profiles, who is the target population? Where does this test need to work? In this case, around a multitude of different settings that have malaria or non-malarial febrile illnesses. Um, who is the population that needs to be tested? Like, is it the severe population? Is it non-severe patients? So, and what is it? What's the action that we want to inform? So we built then, we were able to secure funding based on these TPPs, and we were able then to do trials with potential host biomarkers, but very strictly looking and following what we had laid out in the TPP as the population that needs it and as the target goals that we need. Well, we didn't find the holy grail and the sort of silver bullet that we hoped for, that like a new marker that we could use. So that was maybe disappointing, but it was also quite, I think, important to connect those target product profiles with like real clinical data that we collected in that setting. But as I said, we didn't find the right tool, but we did understand from the TPPs what is it clinicians really need, and they need tools to differentiate. At the time, that was what we understood, bacteria from non-bacterial infections in that broad febrile population. So another way for us was we collaborated with colleagues in Vietnam at the time, and also others around the world, like to use existing tools and see how can they be used, these tools that we don't need to develop, but they exist. They might be imperfect and nobody would sort of put them forward like and say, this is now the thing that saves us, but they exist. In this case, C-reactive protein, CRP. Would this also have impact in some way related, like linking back to what we need from what was defined in the TPP? Now, yeah, again here, we found some ways forward and found some things that worked and some that didn't work, but it was really impactful to do that in a real life setting. So not in the lab, not researchers that only do that all day long, but like normal clinicians and normal lab people. Now, what am I taking away from like this experience? What did I take away? One of the main things for me was, and maybe something that is important for everybody, like technology alone didn't really solve the problem. Like there's a lot more to it. The infrastructure, the question that I was always asked mostly when I was trying to advocate for better diagnostic is who will pay for that? And that was kind of really like sad, but it's an important aspect. I think if we want impact, we need things to be used. They can't just be cool. And I realized also like being in the room is maybe sometimes more important than having the best data and the most coolest study. Having a seat at the table, you need to have evidence, yeah, for what you're trying to advocate for. But I found that was an important learning for me to understand that coming from this really stringent science background from my lab, that having evidence is one thing, but then there's a big part of shaping how you can have impact and how you only have impact with diagnostic tools if they're used and if people trust the results. And that requires a lot more than just having the test and maybe a cool idea of a new test. Like I kind of thought it would be like that. And yeah, so maybe the problem wasn't the test all along, and it was kind of more the system that we all operate in. Um, and and I think that was, yeah, I think that's the sort of takeaway for me with from yeah, transitioning from the lab to like a more strategic role, that we need both, but we need to think about the end in mind. And so I'm trying to now implement that in my research going forward with this is like an ongoing project right now um, where I'm trying to sort of internalize, where we've tried to internalize, or I've tried to internalize this learning, like really alongside the technology development, which um, is ongoing, to really work on the implementation side of things. I think people that might have joined last week, they heard from Daniela about implementation research. And it's, yeah, about identifying these barriers, facilitators, like what do we need in terms of infrastructure, regulations, people's understanding, people's like, yeah. And then what evidence do we need to convince? Maybe that's also something we need to understand, and what kind of tools are they so that when we have that alongside the new tool, we can then, I think, come up with a good plan to actually implement it, because only then can we have the impact that we hope it will have. Otherwise, we only have maybe a cool tool. This is branching into a new area for me, going into taking sort of what I've learned in infectious diseases and exploring that in non-communicable diseases, neurological diagnostic. So let's see how that will go. We're quite excited, and I'm also quite excited to learn and see. This is the first time I think where I'm really part of a project where we're starting in parallel the two things. And yeah, let's see how that really helps getting impact and maybe positive uptake in the end. Let's see. One other kind of, even zooming out even more, like talking about infrastructure is one thing, but there's everything is so connected. And when we talk, particularly when we talk about febrile illnesses, it's anything from dengue to malaria to rickettsial diseases, leptospirosis. Lots of things could cause that, and a lot of them are not like they're very much cholera, like they're very much related. Well, currently Ebola, if depending where you are, very much related to the infrastructure around you and all the SDGs. And the interconnectedness of that is really something that has kind of captured my interest, because how can we work on one little thing and trying to implement that, even if we have the guidelines and even if we have a beautiful lab, if everything around it is kind of crumbling and not sort of suitable, like in terms of infrastructure, in terms of partnerships, in terms of funding, in terms of, yeah, access to clean water, all of the SDGs. So this was work that we did in collaboration with colleagues in Pakistan, specifically also around typhoid, and it kind of brings that story home, like looking at published literature and all the challenges that I mentioned in relation to diagnostic, but how they actually relate to a lot of other aspects that we have in the SDGs. Um, yeah, and maybe sort of this is my real yeah, the learnings I've sort of taken away from my research journey in diagnostics so far. Yeah, I've already said it, like you need to understand what's causing the disease. If you don't know what makes people sick, you're basically flying blind. So if you don't have tools, if we don't have good surveillance, if we don't have any of those things in place, and maybe sometimes they don't need to be the perfect new tool, but they just need to be used. There need to be tools that are used. So understanding really what's out there, and I think, yeah, we're getting better at understanding that. COVID really did was good for everybody to kind of get diagnostic into people's heads, but there's lots of other lots of other pathogens, lots of other conditions that require improved diagnostics so that we actually know what we're dealing with. And then we can also develop the cool tool. But the cool tool alone is probably not going to be it. We need to think when we know what's causing illness and what's causing issues in populations, we need to define what is it really we need in terms of diagnostic. So the target product profile, there's other terminologies around the preferred product characteristics. If that interests you, there's a whole database, a whole website, and loads of websites at the WHO on the WHO or websites dedicated to it. So I think this is kind of we need to define what we need so that what is developed is really also meeting the needs later on, and so that we can also get people's buy-in into the tools that we need. Because if you can't get them used, what's the point? I mean, thinking about implementation questions, who is using it? How complicated can it be? What's the temperature there? Is really essential from the get-go. And yeah, I'm a sort of very pragmatic person, so I think sometimes maybe using an imperfect tool, and some of the studies that we have done show that like maybe we didn't have the impact as huge as we had hoped, as in terms of maybe reduction of antibiotics, if that was the measure. But you know, incremental change is also change. So I think that is something we need to maybe also be open to, and particularly after using things in the real world and not just sort of under very controlled lab conditions, we have to accept that there is some reduction of impact. Um, yeah, we need to educate people to work across disciplines. I'm coming from a lab background. I have an engineering degree from a German university from a long while ago. I've worked in public health. I worked in international organizations. What I've really seen is that we need to kind of be able to talk across the disciplines, which I think is exactly what GLOHRA is trying to achieve. Because in order to do what we're trying to do, maybe with where we try to talk to the technology experts about a technology that they're developing, but we need to translate the issues that we see in the field, or whatever you want to, in the reality, in day-to-day life. We need to be able to translate that in language that is sort of, yeah, understood across clinicians, lab people, engineers. So I don't know, everybody that's involved needs to kind of step back and be a bit humble and kind of try to understand the other people. And I think by working interdisciplinary, we learn that. We learn to really listen to each other, and not accept that we are the only person that knows what's happening. And yeah, that's maybe, I think, sort of a big broader, and maybe I thought for this audience might be interesting. That's sort of a big what I've sort of taken away from my work so far. Yeah, build networks. A lot of the really cool stuff I would have never done if I didn't know the people, whether that's like detecting, being part of a team working on a pathogen in a context that we didn't know, working in countries where I thought I would never work in, or yeah, or do this like just sort of be part of networks, and harness those connections. Really, maybe not only spend the time in the lab, but also spend it building friendships, because ultimately, yeah, that's what kind of brings you to a cool project, and that what brings you to the end of a project and being excited about like a new one. So that's, I think, what's really important. Yeah, don't be afraid to be the only person that like be one of the people that maybe doesn't belong very heavily into one discipline, but sort of bridges. It's not very popular in Germany. It's not a very, I think at the moment, it's not yet a very thought after skill, but I do think it's really important to solve global health problems, and because we need to see and think a little bit bigger and be creative. So that's, yeah, I think something that I feel quite strongly about, and hopefully others do too. Yeah, looking forward, because this was sort of a story from the past a little bit, not yet, not all of it, but yeah, I think it's really, um, I'm still really keen to work in and develop and be part of teams that develop cool solutions. But I also want to be sort of a bit the voice that of reason and say, okay, who's going to use this? Who's going to pay for it? How are we going to do this? Who are we going to implement this? So I think that's something that I'm very much looking forward to, but stay connected to problems and solve problems, public health problems like, yeah, aging populations, zoonotic challenges, so on. Yeah, work with new and old friends and collaborators. If you spend like a couple of years working on a project, hopefully you end up being friends. Um, that's a great advantage of I think our job. Yeah, or get in touch if you're interested in collaborating on anything. I do a lot of education now, and I like it because I think we need people that think outside the box. So maybe contributing to that a little bit. And we have like local challenges in Bavaria, like little zoonotic critters that require support in surveillance support. So we're working with the Bayerischen Landesamt für Gesundheit und Lebensmittelsicherheit, LGL, in Munich on like an interesting project, sort of supporting their surveillance for Borna. If you haven't heard of Borna and you're in Bavaria, no, that's impossible. Everybody in Bavaria has probably read about it, but if you're not, Google it. It's quite an interesting case of a very deadly virus that we seem to be particularly, yeah, we have cases here where nobody has them on otherwise. So that's really a cool little project. Not that little, but a little um, shrew here shoe-related project that is going to be fun and contributing, hopefully, to understanding that question that I started with, like where do we find the things that make people sick? Yeah, and that's sort of the end. Like these are where I've worked, where I've studied, and or where I did my PhD. And yeah, I guess all the places and all the people that shape you. Or shaped me. Yeah, and I couldn't not like do some advertising. If anybody is looking to do a Master in Global Public Health, join us. We are very lo rural, yet very, very international place. So if anybody finds that kind of intriguing, get in touch or apply. Yeah, that's the end from my side. Thank you very much for listening.